So when I talked about the cytoskeleton, I really focused almost exclusively on microtubules. And that's partly just a comfort thing. I've always found actin and its control rather intimidating because there's so many actin associated proteins and I can never remember what they all do. And I don't really expect you to either, but it's important to understand some fundamental aspects of how actin works within cells, how it's controlled. And I'll talk very tangentially about how it's leveraged by pathogens, sometimes to enter and move around cells. So this is just the same introductory slide I used like a week and a half ago when I talked about microtubules. I guess it was 2 weeks ago. And it just illustrates the fact that actin is the other major family of dynamic polymers within cells. Intermediate filaments are not as dynamic because they are not enzymes. Both microtubules and actin filaments are comprised of subunits that have, in the case of microtubules, GTPase, and in the case of actin, ATPase activity. And that enzymatic activity helps to give them the ability to rapidly assemble and disassemble in a highly controlled way within cells. Actin is probably more, well, I find it more confusing than microtubules, which is why I sort of skipped it. But today I'm going to try to tell you what I think are the key things to know about actin. This just illustrates again how microtubules can very rapidly assemble to form mitotic spindles. This is again a Drosophila embryo undergoing these rounds of synchronous cell division. Actin is probably best known for its role in cell movement. And I'll show you some other movies of cells like this. This is called a keratocyte. It's a cell derived from fish scales. And they're used because they have this very beautiful linear motion that you can recapitulate on a cover slip and watch in vitro. Okay, so actin takes on many different forms within cells. And it polymerizes in different ways. And that's one of the things that I think is intimidating about it, is just understanding all the different ways that it can be put together. But ultimately, it really has two forms. The filaments have two forms. They can either be branched networks that grow off of the sides of each other, or they can be straight. And I will show you how those two different kinds of actin networks are assembled in cells. This just illustrates different places that actin can be in cells. It can be in microvilli, and I'll show you how it's sort of bound together to give these structures some rigidity. It's often concentrated near the plasma membrane. If the cell isn't moving, that kind of concentrated region of actin is just referred to as the cell cortex. And when cells are moving, they form various structures like lamellipodia and filopodia, which are defined by kind of their overall morphology, and I'll show you some images of that. And then we talked about how actin is involved in cytokinesis and pinching off cells as they're dividing. And we talked a little bit about how actin can be induced to push cells into a polarized morphology. In the case of these yeast cells, they're schmooing in response to a pheromone, which induces them to grow toward a cell that they're going to mate with. And then really, one of the first well-studied functions of actin in cells is in muscle contraction, and I'm largely going to skip that because I think you can, with the principles that I'll present today, you can definitely understand what the book has to say about how muscle contraction works. All right, so this is just another version of what I just told you, showing different networks of actin in different places in cells. I didn't mention these. These are called stress fibers, and they're often seen in non-moving cells, and they are part of what gives the cell overall rigidity and strength. We often visualize actin in cells using a really useful reagent called phalloidin, which I'll show you in a second. Actually, I'll come back to that. So this is just a table in the chapter that lists, as I talked about for tubulin, there are a number of naturally occurring compounds made by different organisms for reasons we often don't understand that when purified and added to cells do interesting things. So I talked in the case of tubulin about Taxol, which stabilizes microtubules, and colchicine and vinblastine, which destabilize microtubules. Similarly, there are small molecules that can stabilize actin filaments, and small molecules that inhibit the assembly of actin filaments and thereby destabilize them. And phalloidin has, so phalloidin is a really interesting compound. It comes from this mushroom called Amanita phalloides. And actually this mushroom is called the death cap, and it's responsible for most of the fatal mushroom poisonings in our area. So if you ever see big white mushrooms with white gills, you should not eat them. And so it's a natural toxin. It's actually not, the phalloidin is not actually what kills you in these mushrooms. It's the amanitin, which inhibits protein synthesis or RNA synthesis. I always forget. I'll come back to that. So phalloidin, though, is also toxic. It's not as toxic as amanitin, but it binds. So the useful thing about phalloidin is it's a compound that binds to the interface between actin monomers in a filament, and that's how it stabilizes the filament. And the nice thing about that mode of binding is because it only binds to that interface, when you use it to stain cells, it'll only stain the filaments. It doesn't stain the monomeric actin. So it's a really useful tool for cell biologists to visualize actin filaments. By itself, you can't see phalloidin, it's not fluorescent, but you can chemically link it to fluorophores like rhodamine and add it to cells. This is the structure of rhodamine. And then you can see where all the actin filaments are. And this is just to know, I use this slide in 133L where we teach more about fluorescence, but I just want to highlight the fact that, as I said, the phalloidin is not fluorescent, but you can link it to a variety of different fluorescent dyes, and the fluorescent spectrum that you see in the microscope is determined by this dye, not by the phalloidin. Okay. So, as I said, both tubulin and actin proteins that make up the filaments are enzymes, and tubulin, beta tubulin is a GTPase, actin is an ATPase. And very similar to tubulin, in the ATP bound configuration, it is more inclined to polymerize, to bind to other actin subunits and thereby to form a filament. But in the filament, it hydrolyzes ATP, and the ADP bound form is less stably associated with the filament, and so it will tend to disassemble. And so in vitro, you can get a situation like this where actin can be simultaneously adding to one end of a filament and dissociating from the other end of the filament in this process called treadmilling. In practice, although in cells there can be sort of steady state accumulation of actin and disassembly of actin, you don't see a situation like this, because in general, the ends of actin filaments in cells are not free. They are capped by actin capping proteins, which I will show you. All right, so I think I should maybe skip over this in the interest of time, but I'll just show you because it's in every textbook, and so I'll just mention it. And I showed you something similar for tubulin, that in vitro, if you take a concentrated solution of actin bound to ATP, it will, at high enough concentration, it can spontaneously assemble into filaments as long as you're above a critical concentration. And it's known that there's time required, there's an energetically demanding step of nucleation for the first few monomers to come together to form what's called a nucleus. And once that happens, then you get sort of a steady state of actin incorporation until the amount of free actin that's not incorporated into filaments reaches this critical concentration, and then you get a steady state of addition and removal. Like I said, that is not really what happens in cells, because cells carefully modulate where and how much actin polymerization happens using a wide variety of different binding proteins. So that I'll show you what I mean by that in a couple of slides. But one way is that the free concentration of actin in cells is not equal to the actual concentration of actin in cells, because the monomers of actin are actually bound by monomer binding proteins that control whether or not they can assemble into filaments. Okay, so actin is polarized, and so are microtubules, and that's important because it allows motors to walk in a directional way along actin filaments. And it also allows directed assembly and disassembly of networks of actin. Sometimes people use this trick to visualize actin for electron microscopy, where they take a little fragment of myosin. So as I'll talk very briefly about, myosins are a large family of motor proteins that walk along actin. And it was discovered many decades ago that if you take a small fragment from one of these myosins, it will bind to the actin filaments in a way that gives this barbed appearance in the electron microscope. And so people realized that the plus end was equivalent to what they call the barbed end when the actin filaments are decorated with these myosin fragments, and the other end is called the pointed end or the minus end. And again, the plus end is where ATP-bound actin will preferentially assemble, and the minus end is usually where the actin filament is growing from. But in practice, these ends are not free in cells. Usually the pointed end is associated with either a capping protein or a nucleator, and the plus end is also associated with capping proteins. Okay, so these are different roles of the asymmetry of actin filaments, which I've mostly mentioned already. Okay, and that's just a restatement. So now I'm going to move on and start talking about this kind of huge constellation of different actin-associated proteins. And I have a hard time remembering these, and so I have kind of a mental block in dealing with actin. And I think it's best if you don't try to memorize exactly. I mean, you have to know what some of these things do, and you have to know them by name. Ooh, someone's drawing on my slide. But mostly I want to kind of drive home, you know, how these things regulate what actin does in cells. All right, this is another version of the slide that I just showed you, but it has actual kind of molecular structures. But I'm going to go through what I think are a few of the key functions of actin binding proteins and how they work. All right, so this is just to illustrate kind of what's happening in a migrating cell. So as I said, the cells actually I didn't really say, but cells push themselves forward if they're kind of crawling along a substrate by polymerizing actin at what's called the leading edge of the cell. And in most cases, the leading edge will have this kind of flat appearance that's called the lamellipodium, and it's full of polymerizing actin. And as the cell is crawling this way, it's establishing contact with the substratum, which is usually extracellular matrix if you're talking about in an organism. And we talked a little bit about how connections form between the actin cytoskeleton and extracellular matrix through the action of integrin proteins. Of course, in order to move, the cell has to not only polymerize actin at its front edge, but it also has to, in a kind of balanced and regulated way, release the contacts and contract the minus end, not the minus end, the back end of the cell, which happens through, in large part, through the action of myosin motors that are sliding along actin filaments in the back of the cell. Sorry, I have to. Okay, so this just shows another image of the leading edge of a cell, and the main kind of crawling edge, as I said, is usually this kind of flat region that's called the lamellipodium. Some cells also have filopodia, which are just thinner projections of actin and membrane that enable them to kind of feel around and assist in movement. But most of what I'll show you is lamellipodia. So I showed you a cell like this earlier. This is a keratocyte crawling along the surface. And this is the same type of cell, but in this case, it's been labeled with GFP actin. And well, I'll describe what's going on here in a second, but this kind of allows researchers to actually study what happens to actin filaments as the cell is moving forward. And the reason that they can do that is that in this case, they're using something called speckle microscopy, which means that not every actin molecule in the cell is labeled. It's kind of patchy. And so in the microscope, sorry, I'm going to play that again, you can actually track kind of individual little regions that are bright and dark, and you can see that they're not that they're not individual actin filaments are actually staying put. There's growth at the edge of the cell, and there's retraction of actin at the back. And that is what enables the cell, that's kind of treadmilling behaviors, what enables, oh shoot, is what enables the cell to move forward. Can you still hear me? Because I just got a message that my sound is off. Kirsten, can you hear me? Okay. And my children have just opened the door, so I'm going to go close the door. All right, good. Hopefully you can continue to hear me. All right, so the other thing that movie showed, sorry, there's a lot going on in this movie, is that at the leading edge of the cell, you can't see it by light microscopy, but we know from electron microscopy that the actin at the leading edge of the cell is in this branched network. And I'll show you other images to highlight that, and then I'll describe how those networks form. So it's a little hard to see here, and probably on your screens it looks really crappy. So I'll show you another, I'll show you some better images that are not moving, so they're better. Okay, so this summarizes what I already told you, that within a cell, this is all being highly controlled. It's not just spontaneous actin polymerization. The effective concentration of actin within cells is controlled by actin monomer binding proteins. And I'll show you particularly the role of profilin in actin polymerization in a sec. And all of that network of actin polymerization at the front of the cell is driven by nucleation factors. So this is the kind of slide that I hate looking at because it's got too much information, but this illustrates a couple of the different actin binding proteins that play roles in modulating the activity of actin. Thymosin by essentially sequestering G-actin, making it unavailable for addition to filaments, and profilin doing sort of the opposite, binding to actin in a way that it can deliver it to the growing edge of a filament. And that should become clearer in a second. Okay, so this is kind of what this network at the front edge of the cell, the crawling cell that I just showed you, looks like. And this is reconstructed through cryo-electron microscopy and other techniques. So in this image, the orange proteins are the actin monomers, and they are kind of stacked together in this sort of helical structure to make the actin filaments. And I mentioned in my previous cytoskeleton lecture that actin filaments, unlike microtubules, are not hollow. They're just kind of solid but helical rods of actin. And if there's time at the end, I'll talk a little bit about why they have that kind of organization. And what you're looking at here are actin filaments, and then this nucleating complex called ARP23. ARP is actin-related protein. So it has a structure similar to actin, but it binds to actin filaments, and then it gets activated by another protein that I'll tell you about in a second to catalyze the nucleation of a new actin filament growing off the side of an existing filament at a 70-degree angle. And that helps to create this branch network that has, so actin, as I mentioned in my previous lecture, is quite flexible. Microtubules are very rigid. Actin is very flexible, but by growing in this branched way, the actin network can create enough kind of mechanical strength that it can push the membrane forward and execute this kind of movement. And this is just another image of actual data showing how people can develop ways to kind of peel the membrane off of a fixed cell and shadow the actin network with metal so that they could look at it by electron microscopy. Okay, and here's another image showing the 70-degree angle between one actin filament and another actin filament growing off of its side, and how that leads to this branch network at the edge of cells. Okay, so I mentioned that ARP23 is the key nucleator for these branched actin networks, but it doesn't act by itself. it requires an activator from this family of proteins, the first of which was was called WASP, for Wiskott-Aldrich syndrome protein, because it's mutated in this disease. So I always have to relearn this every time I teach it. So if you have to think about it for a while, do not feel bad. But the way that this whole system works is that ARP23 can bind to the side of an actin filament. By itself, that will not efficiently nucleate a new actin filament. It needs this activating domain and this profilin binding domain from this other protein called WASP. So WASP, when it's floating around the cell by itself, is inactive. There's a large family of proteins like this that are important for actin polymerization, and they all have a domain. In this slide, it's called a row binding domain, but more generally, they're called GTPase binding domains, or GBDs. And these domains typically inhibit WASP and its family members and other actin polymerization factors unless they are bound to an active form of a GTPase. So we've learned about a variety of different G proteins or small GTPases, some of which are attached to the plasma membrane of cells. So this is one of them. We haven't talked about this particular one, but it's part of a family of GTPases related to Rho and RAC, and this one's CDC42. And so in this diagram, what it's showing is that in the presence of active CDC42, so the GTP-bound form of this G protein, this RBD domain will bind to CDC42, and that will relieve the inhibition of the rest of this WASP protein. And it will do a couple of things. It will enable this domain to deliver actin. It's actually actin bound to profilin, although that's not shown here. And it will also induce a conformational change to the ARP23 complex that promotes the assembly of actin. So it's very highly regulated, just like everything in cells. Okay. And this shows an in vitro assay using ARP23 and WASP to show that in vitro you can nucleate these branched actin filaments. I'll just mention, because it's on the slide, that this is using another microscopy technique called total internal reflection fluorescence, which is really useful for imaging this kind of cytoskeletal assembly in vitro, because it's a technique where you use this kind of special illumination that just illuminates a very thin area right next to the cover slip. So you don't see all of the fluorescent actin that's floating around. You only see the polymerized actin that's right next to the cover slip. Oh, okay. Kirsten is transcribing what I said, which is weird. All right. Okay, and I mentioned that the free ends of actin filaments are generally not free in cells. They are, as actin filaments are growing, whether they're growing nucleated by ARP23 or other nucleating factors, as they're growing, they rapidly get capped by different capping proteins, so just generically called capping proteins here. And the capping proteins keep, so I showed you that movie just now where the filaments were growing super long in vitro, and that would not be very useful to cells because they would be too flexible to actually execute any movement. So by nucleating these branch filaments and capping them quickly, the cell generates these kind of short, highly dense meshworks of actin that have structural rigidity and can do work. And this is just a couple of examples of capping proteins. You do not have to memorize them, but just to illustrate that there are both plus and minus end capping proteins that have different functions. Okay, and this is basically an illustration kind of putting together everything that I've told you so far about the nucleation and function of these branched actin networks at the cell surface, that they're driven, so that the nucleation is promoted by active G proteins at the plasma membrane that activate WASP or other family members, and together with ARP23, they drive assembly of branched actin filaments, which are quickly capped by capping proteins. Okay. So people were aware of ARP23 for a long time, and more recently, not so recently now, but more recently, it was discovered that there's another whole family of actin nucleating proteins that behave in some ways a parallel way, but in some ways a rather different way. Whereas ARP23 are found at the minus end, the non-growing end of an actin filament, there's this family called formins that surprisingly turned out to be associated with the growing end of an actin filament. And they work not so differently, but what they do is they coordinate the addition of actin to the growing end of an existing filament. And so they nucleate straight actin filaments rather than branched ones. And I'll show you this little animation, so how this formin protein is thought to track with the growing end of an actin filament or as it retracts. And this is, again, in vitro assay showing like that actin by itself will, I don't know if this, yeah, so this actin will grow slowly by itself, spontaneously, in the absence of formin, but in the presence of formin plus this actin binding protein called profilin, the rate of growth is much greater. Okay, so there's a lot of similarities between the way formins work and the way that ARP23 actually WASP works. So formin, like WASP, has a GTP binding domain, or in this case, it's again labeled Rho binding domain. And it also has these domains. There are up to three different domains that are called formin homology domains that interact with different aspects of actin and actin regulation. In this case, it only shows formin homology 1 and formin homology 2 domains, and it's showing what they do. So the formin homology 2 domain allows the formin protein to remain associated with the growing end of an actin filament. The FH1 domain in this case binds to profilin actin and helps to deliver it to the growing end. And as with WASP, the GTPase binding domain acts like an inhibitor of these other domains unless it's bound to an active GTPase. Now, I haven't really described what profilin does, but profilin acts, it enhances binding of ATP to actin. So it acts a little bit, so we've talked about guanine nucleotide exchange factors that promote release of hydrolyzed GDP and binding of GTP to G proteins. This domain acts similarly with actin. It promotes actin's release of ADP and binding to ATP, and thereby promotes its incorporation into filaments. And it's also what is recruited by this FH1 domain to deliver actin to the growing end of filaments. Okay, so there are different ways that actin can polymerize, and there are different G proteins in cells that have different effects on actin polymerization when they are triggered. And I am not going to get into this at all, but this just shows images of what happens when Rho is activated, when CDC42 is activated. So here you see stress fibers. Here you see more actin polymerization into, I guess these would be filopodia at the edge of the cell. And this looks more like, I guess, lamellipodia, or I'm not really sure what's going on here. And this is an unstimulated cell. So let's see how we're doing. Okay, so as you know, GTPases can be activated by extracellular signals, and typically are. That's kind of what's shown here is different signals that have been added to cells to activate these different small GTPases or monomeric GTPases. So that's a major mechanism that controls actin within cells, is that there are signals that come in and activate one or more of these small GTPases. This shows a different mechanism of activation. In this case, through a G protein coupled receptor, you can get, and we talked about this kind of pathway before, where a G protein coupled receptor can trigger downstream second messengers, sometimes through activating enzymes that produce those second messengers, in this case, inositol triphosphate and diacylglycerol, which then activate protein kinase C. So protein kinase C then will modulate actin activity by phosphorylating many other actin regulatory proteins that are way beyond what I'm going to talk about today, but they're analogous to the MAPs that I talked about with respect to the microtubule cytoskeleton. So these are proteins that can bind to actin filaments and affect their behavior. Their fashion, as you might imagine, is a bundling protein that so activation of different proteins by these global regulators of cell physiology also have major impacts on the actin cytoskeleton. Okay, so I mentioned that one reason I decided to talk about actin is because actin is often used by pathogens for their own purposes. And I'll point you to a really good review on this topic. One of actually two of the leading labs in this area are at Berkeley. One is Dan Portnoy's lab and one is Matt Welch's lab. They both study how pathogenic organisms kind of modulate the actin cytoskeleton and do things with it. And I'll just show you a few examples, and I'll post a couple of papers that you can read if you're interested in more details. This is an example of E. coli in the gut, which I don't know exactly what they're doing here, but they make these structures called pedestals. So they trigger actin polymerization to make these weird like landing pads on the edge of cells. And I'm not quite sure exactly how that helps the E. coli. Salmonella, which is also a pathogenic bacterium, actually induces its own uptake into cells by triggering what's called phagocytosis, where the cell kind of using the actin cytoskeleton, it makes these ruffles, and eventually those will lead to the cell actually engulfing the bacterium and taking it internally. And these are very common kinds of themes. These are examples of how bacteria, in this case, use signals, external signals, either to interact with proteins on the cell surface, or they'll actually inject signals into cells that trigger rearrangement of the actin cytoskeleton. So this is kind of a cartoon of what I just showed you in this image, where Salmonella is actually injecting effectors or signals into the eukaryotic cell through what's called the type 3 secretion system, and that triggers the polymerization of actin into these ruffles that eventually will eat the bacterium and help it get into the cell, not destroy it. There are other cases where bacteria express what people call zipper proteins, but basically they are proteins that interact with proteins on the cell surface that then lead to engulfment of the bacterium. And this is very analogous to what I talked about with coronavirus, which is a virus, obviously not a bacterium, but coronavirus interacts with a cell surface protein, ACE2, that somehow, we don't actually know exactly how, helps to trigger its uptake into cells, either by clathrin-mediated endocytosis or by a non-clathrin dependent mechanism. So, and there are many kind of variations on this theme. This is kind of one of the simpler versions that I was able to find to show this kind of behavior. And then we also know that a lot of the early studies actually of actin nucleation were spurred in part by the discovery that many bacteria have their own actin nucleating proteins that enable them to use the host cell actin to move around within cells. And that's what's shown here. This is an infection of a cell with a bacterium called Listeria monocytogenes, which causes listeriosis. And what I'll show you some movies, but it turns out Listeria expresses a protein called ACT-A that it localizes to one side of the cell, to one pole of the cell. and it enables actin to be nucleated and drive the movement of these bacteria around the cell. And also, eventually, it helps the bacteria to escape a cell and go into a neighboring cell by providing force and movement. I just mentioned here that listeria is not generally a very serious disease unless you're immunocompromised or pregnant. For reasons that I don't know much about, infection with listeria can cause miscarriages, and it can also cause meningitis in newborn babies. So that's why if you are pregnant, you are usually advised not to eat cheese made from raw milk, because that can be a source of listeria, as can, for some reason, cured foods like salami, luncheon meats and things like that are usually advised against for the same reason. But actually the main source of listeriosis, as far as I know, is often through air conditioning systems. Often there'll be like a cluster of listeria cases that people all got from an air duct that was contaminated. So this illustrates how listeria moves around inside the cell. It uses the host actin and the host ARP23 complex, and I think that this Act A protein takes the place of the WASP. Oh yeah, that's what it says. It behaves like WASP, so it activates ARP23 to polymerize these branched actin filaments behind it, and that and the addition of actin right here at the back end of the cell actually propels the bacterium forward within the cytoplasm of the eukaryotic cell. So here's some movies of that happening. This is a phase contrast image of listeria kind of rocketing around the interior of a eukaryotic cell, and you can see sometimes that it makes these projections. It's trying to get out into the extracellular space. But if this cell were in an organism, those would also enable the cell to invade adjacent cells. And I think that might be shown here, although it's not a very clear movie, but you can see again these projections coming out of the cell and helping the bacterium to spread. So again, Matt Welch's lab was really a pioneer in elucidating how listeria and this Act A protein that it makes can polymerize, can help drive actin polymerization to move listeria forward. And more recently, they have also worked on other bacteria that use other actin polymerization systems to drive bacterial movement within cells. So this is a different family of bacteria, Rickettsia, that's known for causing Rocky Mountain spotted fever, and I think maybe other diseases. They study a non-pathogenic species of Rickettsia, and they showed that it actually polymerizes. It has a protein called SCA2, which behaves like a formin and polymerizes straight or unbranched actin networks, but they also have the effect of creating kind of comet tails within the cell. And so I'll post this review because I think it's really nice. It's not super recent, but it's a nice review with a couple of really nice figures. And I'm not going to read all this, but it's basically a summary of what's shown over here, that different bacteria kind of harness, they take advantage of the host's actin polymerization machinery in a variety of ways. In some cases, they mimic, I showed you some slides when I talked about phase separation. I showed you this network of proteins that polymerize actin in podocytes, including Nick. So some bacteria express proteins that recruit Nick, and that recruits host, NWASP, and so forth. I showed you what Act A does, that it acts like NWASP to activate ARP23, and then that Rickettsia has this protein that acts like formin. And there are many other variations. Okay. And yeah, so I just wanted to illustrate a few other kinds of actin-associated proteins that do important things. Like I said, I'm not big on memorization, and there's a zillion of these proteins. I think actin interacts with directly interacts with at least 100 different proteins inside cells. So it's like impossible to remember them all and what they do. I've tried to highlight what I think are the key ones. This is a couple of examples of cross-linking proteins, fibrin that can bundle actin fibers together to create much stronger kind of bundled fibers. Fashion, I think, does something very similar, as does actinin. So these are expressed in different cell types with different functions, and I don't remember what they all do. It's just to illustrate that there are different ways to kind of bundle these filaments to create different structures with different kinds of biophysical properties. And many of these proteins are linked to diseases. Okay, this is just a blow up showing that this structure is what's inside these microvilli in cells in the intestinal lumen. Okay, and then the motors that walk along actin filaments are all related to each other, and they are members of the myosin superfamily. So these are proteins that have either one or two head domains. Well, they have one head domain, but they can dimerize to form essentially a two-headed protein. And they undergo a cycle of ATP hydrolysis that enables them to do movement. And so they grab hold of an actin filament and they hydrolyze ATP. And I think it's actually the release of ATP that drives the main conformational change that drives movement. This is the structure in a muscle cell of the myosin. So you are probably aware that there are kind of intercalated actin and myosin filaments in muscles that contract relative to each other and drive muscle contraction. So myosin 2 is probably the best studied myosin because it's the main motor in these muscle cells. And I'm not going to go through this, but this shows the power cycle of the myosin motors, how binding ATP and ATP hydrolysis and release of the phosphate and ADP drive this cycle of movement and resetting to rebind to actin. Okay, one thing I thought I would just touch on right at the end, because I think it's really interesting, and it's not really in the chapter, but this was something that that I started thinking about after listening to a lecture from Julie Theriault, who happens to be the voice that you have heard narrating a lot of the movies that I've shown you from the textbook. So she's a very prominent cell biologist. She happened to be a classmate of mine in graduate school, so I know her personally, and she's now a director of the Allen Cell Institute in Seattle. And she is really good at kind of drawing together common principles in what seem like disparate phenomena. And one thing that I hadn't really thought about until I heard her talk about it was how does this happen that you have proteins like GTPases or ATPases that form filaments? How does that come about in evolution, and why is that? Why is it that these filamentous proteins are derived from enzymes? So, the why is already sort of evident from what I've talked about, that the GTP hydrolysis cycle or ATP hydrolysis cycle can be utilized by the cell for regulation. So energy can be used in cells in a variety of ways, right? To generate force or other kinds of work, or regulation. And in this case, in both of these cases, you have enzymes that contribute to their own assembly and disassembly and are thereby very useful to cells. And they are arranged, interestingly, maybe in helices. It's more obvious in the case of actin. So I've described that actin proteins, so it's just a single subunit, and they're stacked together in this array that gives you kind of a helical filament. But microtubules are a little bit more complex, but they are also sort of a helical structure that arises from stacking together this dimer of very similar alpha and beta tubulin. And that might seem kind of weird, but it's actually a very, it's expected. And this is what I mean by that. So this is an essay that Julie wrote that I found very thought-provoking. So what she highlighted is actually some ideas that were prevalent in the mid-20th century, that if you have a protein, so a protein is a blob, but it's typically, it doesn't have mirror symmetry, it's an asymmetric blob. And if if this protein has a surface that can bind to itself, right? So if this surface likes to bind to this surface, what will happen eventually is that at high enough concentration, this protein will make some kind of helical structure. That's just like what happens if you take an asymmetrical building block that sticks to itself in some place, that it will naturally make filaments. And in most cases in the cell, proteins that are present at high concentration have evolved specifically so that they don't do this. They don't stick together, otherwise it would be toxic. And I'll show you an example where it is toxic. But presumably at some point, an ATPase that looked like actin and a GTPase that looked like tubulin developed this ability to form helices, and somehow the cell started to be able to harness those helices to do something useful. And presumably that's how these filaments arose during evolution. And this, I think, is a really nice kind of illustration of that principle. So I talked about this at office hours a couple of weeks ago, but I thought it was worth talking about more broadly. So this is a case of an unintentional filament but it happens to cause a disease. So this is hemoglobin. And normally hemoglobin, which is the protein that carries oxygen and iron in cells and in the bloodstream, is not a filament, but it's present at extremely high concentration in red blood cells. So like 300 mg per mil is a like mind-blowingly high concentration. And so this protein has evolved to not stick to itself, because at that concentration, even a very weak interaction with itself would cause a problem. And that's exactly what happens in sickle cell anemia. So sickle cell anemia is a prevalent disease, especially in African populations, because it actually, heterozygosity for this mutation actually seems to confer some kind of resistance to malaria. And so in places where there's a lot of malaria, this mutation is more prevalent. In heterozygotes, it's usually tolerated, but in homozygotes, it's very bad. And so this mutation is a single amino acid change on the hemoglobin protein that causes it to stick to itself a little bit. And the result is that it can assemble into this kind of very large filamentous structure that looks very much like a tubulin, a microtubule, essentially. It's a hollow structure with 14 protofilaments. So this just sort of illustrates that, that in evolution, it's not surprising that a protein, whatever it used to be, that it developed this ability to form filaments. What we don't know is how the cell, started to harness the ability of the tubulin ancestor or the actin ancestor to actually do work. And it used to be thought that this was something that had happened in eukaryotic evolution. It actually was not recognized until fairly recently that prokaryotes actually are the origin of some of these filamentous proteins. And I'll show you that in a second. But actin, it turns out, is structurally related to a variety of other ATPases. And so we imagine that some ATPase that had some other function within a cell developed this ability to form filaments, and somehow that became useful. And like I said, so we used to think that eukaryotes had this cool cytoskeleton, and that was one of the things that made them really different from prokaryotes, and that prokaryotes were more specialized in terms of, or more diversified in terms of their chemistry, their metabolism, their ability to live in different environments and make different kinds of products. But that view has pretty much fallen by the wayside as people have realized that no, actually, these prokaryotes also have filamentous protein structures that do work. It was recognized, for example, that bacteria express this protein called FTSZ, which is a major player in bacterial cell division. And it was found to be a GTPase. It had very not obvious sequence similarity to tubulin, but it turns out that it is almost certainly an ancestral relative of tubulin, because like tubulin, it assembles into filaments in a GTP-dependent manner, and it has essentially the same structure as alpha or beta tubulin, even though that was not apparent from the sequence. So although these proteins have diverged quite a bit between bacteria and eukaryotes, they have probably similar functions. The the array of motors that act on these filaments in bacteria is probably more limited, but they still have filamentous proteins that do work. And likewise, it turns out that bacterial proteins that were called MREB turn out to be relatives of actin, and actually prokaryotes make a variety of actin-related proteins that play different roles in structuring cells and organizing their interior contents. And it was partly, we didn't actually realize that bacteria had organized interior contents because they're so small that until microscopes got good enough, we couldn't actually see that they had any interior structure, but they do. And this shows two of these bacterial actins, MREB and another one called PAR-M, that play important roles in cell shape and partitioning DNA in many bacteria, and they are structurally related to actin. This is just an illustration from Arash Komeli's work. So he's in the plant microbial biology department at Berkeley. And I mentioned earlier that this kind of dogma that bacteria do not have internal organelles bound by membranes is sort of falling by the wayside. They have, among other things, some bacteria have these structures that Arash's lab studies that are called magnetosomes, which are little vesicles packed with magnetic particles that enable bacteria to orient in a magnetic field. And it turns out that the nice, beautiful array of these magnetosomes is organized by this bacterial actin, MAMK. And so this is just some work that showed that. And there are also intermediate filament-like proteins in many bacteria. This is another bacterium called Caulobacter crescentus because it's shaped like a crescent. And this is a really nice model for certain kinds of cell differentiation processes. See that I'm at the end of my time, but I'm also at the end of the slides, so that's good. Anyway, so they have this filamentous protein called crescentin that shapes their cell membrane and gives them this crescent shape. And it's a relative, I don't know if it's evolutionarily related to, but it's similar in structure to eukaryotic intermediate filament proteins. So that brings me to the end of my slides, I think. Let me see.